Got it. All right, well, I'm gonna go ahead and start. Thanks everyone for joining. As you guys know, this is a part of CU Data Week. So this is the, I believe, third day of CU Data Week and the fifth StatsTalk, and the attendance has been so great and we're so excited. So today we're going to be talking about microbiome data and some of the fun caveats that come with working with data of that type. I don't know how you've done it in the past, Lola. Do you do chat for questions or?
I'll monitor the chat for you just so that you don't have to pay attention to that. And then I'll, if you're comfortable with people interrupting, we can do that. Or if you prefer people to wait till the end, whatever is easiest for you.
Let's do chat and then we can answer them at the end.
Okay, sounds good.
Okay. So, like I said, this is Introduction to Microbiome Analysis. I am Kayla. Brief introduction of myself. I got my master's in biostatistics from the Department of Biostatistics and Informatics at CU. And then I worked as a research instructor at CETA until 2 weeks ago. So very fond. Okay, and a special acknowledgement to Brandy Wagner. This talk has been adapted from her talk that she gave on breathable statistics for the CF microbiome at NACFC in 2017, and it's being repurposed and updated for our needs this week. There we go. This is our outline for what we're going to talk about. First thing is just what is the microbiome and why do we care? And then the bioinformatics pipeline that goes with that, and then complications in microbiome data and how do we analyze it in light of those complications, and then some useful packages and pipelines. I've tried to cater this talk a little bit to things that might be a little more relevant for those who are trying to do CU Data Week later this week for the Stats Fest part. So keep that in mind. So first, what is the microbiome? Microbiome is a population of microbes assembled at a particular time in a particular location. The microbiome contains both the microbiota and the metagenomics. The microbiota are the specific microbes in the population, and the metagenomics identify the collection of genes present in the microbiota. Confusingly, microbiota and microbiome can be used interchangeably at times, even though they are not the same thing. Microbiota are a part of the microbiome, but they're not the whole thing. The whole thing also includes the genes. So keep that in mind. So why do we care? The microbiome has been associated with disease states, and the hope is that we can use the microbiome to target diagnostic techniques and intervention strategies. These are things like personalized medicine, probiotics, things like that. I put in here that we also need to understand divergence from normal, because if we don't understand normal, we can't understand what's different. And specifically for things like the vaginal microbiome, we know that there are 5 different types of normal for healthy women. And so understanding what normal means is important as much as understanding what non-normal is. So the bioinformatics pipeline. Before we get to that, we kind of have to think about where do the samples that we're going to run in the bioinformatics pipeline come from. So we want to think about study design. Is it longitudinal? Is it cross-sectional? Who is our population? Is it children? Is it adults? Where are we collecting them from? Is it hospitals? Is it private institutions? Did we send a kit home to them? Those kinds of things. We also want to think about sample collection. So again, did we send a kit home to them? Are they being collected in the hospital? Are they being collected from sputum or a swab? And what are we doing so that we don't introduce contamination into them? These types of things. We also want to think about sequencing region. The data that we get from 16S is not the same as the data that we get from RNA-seq, and that can introduce some different types of bias. RNA-seq is kind of more the metagenomics. It's not the genes associated. 16S does. So this is a, oops. This is a general pipeline for the bioinformatics side. One thing to note is that this is a general pipeline and the different software can vary between pipelines. So I'm just explaining how the general steps so you can see where bias might be introduced and where some of these decision-making things might come from. We're not going to spend too much time on it. We're going to spend more time on the analytics, but it's good to know how it kind of breaks down. So the first part is DNA extraction, PCR amplification, and sequencing. And this is often used, software called Illumina is used to do this. The next and first filter is quality filter. And what we're doing here is we're trying to get rid of multiple barcodes from primers. And this can happen because we're multiplexing, which increases throughput, but also adds bias into our PCR amplification. And so we want to filter out that. And one of the softwares that were used are bar tabs. So basically at the top of each of these little cubes is the name of what we're doing, and then in italics a little smaller is the software that's being used for this. So the next thing in tandem is a chimera check, and we're going to sequence to a SILVA database. So the chimeras are sequences that are formed by 2 or more sequences joining incorrectly. So we want to get rid of those, and then we want to align them to the phylogenetic tree using the SILVA database. Again, SILVA is just one. And then we're going to do a final sequence. So we're going to filter out unaligned sequences and group together the unclassified sequences that we might be getting. And then lastly, we're going to get an OTU table. So as I said before, the pipeline basically agrees in the basic steps, but it differs in the software, and there are some subjective filtering that happens in the final sequence calls, for example. So once you get your OTU table, this is what it looks like. We have on this side right here, we've got our taxa. So this is your phylogenetic tree for that taxa. These are, it just goes down like your normal species call. So kingdom, phylum, class, order, family. And then what we see here are counts from each sample. It's important to note at this point that when we put these data sets together, what's happening is that all of the taxa that sample 1 has is getting combined with the taxa that sample 2 has. So if sample 1 doesn't have one of the taxa that sample 2 does, they're going to get a 0. And it's going to be a 0 either because it wasn't found or wasn't detected. And so that's going to cause some 0 inflation into the data set. And we'll talk about that a little bit more later. So what complications are here? So we just talked about the 0 inflated, which is an artifact of compiling data set from different samples who may have different taxa being found. It's also high dimensional. You can get anywhere from 100 to thousands of different taxa out of these. And often there are small sample sizes, which causes the p greater than n problem. And they're correlated. Remember, this is a phylogenetic tree. So if you look here, these two taxa are coming from the same part of the tree up until the order. They are naturally more correlated than the firmicutes and bacteroides. So that's another fun thing. They're also count data. So they can't be 1.5, they can't be 1.6. They are integers, 1, 2, 3. And they're bounded by the number of sequences for each sample. And each sample might have a different number of sequences from different taxa or from just having more sequences in individual taxa. They're also compositional because they're bounded by the sequences. So if you have 30,000 sequences that you pulled off of your sample, then the number of counts for each taxa lie within that 30,000. It all adds up. And then other things to think about are complex designs, because you can get longitudinal studies of microbiome data and repeated measures, and you have to account for that as well. So why do we care? Well, because of all the things we just talked about, most statistical methods kind of fall apart when we try to look at microbiome data because it's not normal and it's compositional. And if we ignore them, we're going to introduce bias into the parameters and standard errors. So we're going to find spurious correlations and misleading results. This is a really fun quote. Microbial data has been known to present unique statistical challenges that defy most conventional models. That's what makes it fun. Those are really frustrating. So we talked about all of that. How do you actually analyze these things? Well, I like to think of it in 3 different categories. This is kind of the standard as well. So you have total bacterial load, alpha diversity, and beta diversity. For your total bacterial load, this is the measurable quantity of bacteria on an organism. It is not the same as sequence counts. Your bacterial load is what you're cutting from swabbing. So it's like, say you have a cheek swab, you swab, the total bacterial load is what can be measured off of that swab. The sequence counts are how many of that makes it through your bioinformatics pipeline that you've now sequenced and you can count and is in your OTU table. This graph is a really good demonstration of the fact that total bacterial load is not the same sequences. But total bacterial load can still be an indicator of clinical outcomes, and it's often referred to as the total bacterial burden that the host is carrying. The next part is your alpha diversity. So alpha diversity is often thought of as a composition of richness and evenness. So richness talks about how many different taxa do you have. So do you have just a couple taxa, or do you have a lot of different taxa? How diverse are you? Your evenness is talking about how abundant are the taxa that you're finding, and are they similarly abundant or not? So here you can see that this person is not rich or even. They don't have very many taxa, and their evenness is all over the place. Whereas here, this person has a lot of taxa, and their abundance is evenly distributed amongst the taxa. So your diversity is going to be a combination of that richness and evenness. There are a lot of different alpha diversity metrics out there that can be used. The main thing to know is that they are all related to each other. I don't go into the profile curves, but they're basically all related based on this parameter Q. And Q is a really, really great diagram that Brandy Wagner made. Q basically is how abundant each of these taxa are. So if you have a really dominant and abundant taxa and that's weighting most of your diversity metric, then that's going to be up here. These Simpson and Genie and the Renby, those are going to be the ones that highly weight your dominant taxa. Whereas down here, your taxa that are more rare are going to be highly weighted in your Jacquard and your richness. And then kind of the equilibrium where neither the dominant or the rare are being preferred is this Shannon diversity metric. Something to note for people who are doing the stats fest is that the Shannon diversity metric for alpha diversity is the only one that we gave you. We gave you the SOBs, and then also the SOBs is the richness, and then also the Shannon E, which is the evenness. But as far as the combined diversity metric, we gave the Shannon. But there are many more, and you could Google lots of them. They're kind of like covariance structures. So that brings us to beta diversity. So we've talked about the total number and how prevalent they might be, and we've talked about the total bacterial burden. Now we want to talk about, well, how do we put together all of these thousands of taxa that we have? So anyone who's studied statistics a little bit knows that one way to do this is through dimension reduction. And dimension reduction can also be called coordination methods. And what they seek to do is take all 1,000 of the taxa and shrink them into 1 or 2 components. And that is the most variance explained in the data. So we say, okay, well, if I have this one component and it's related to Streptococcus and Pseudomonas, that's driving one cluster versus Haemophilus might be driving another. That's your classical kind of PCA clustering. But for microbiome data, because it's compositional, the things that are upregulated in component 1 are necessarily downregulated in component 2. Because if you have a lot of strep, you can't also have a lot of Haemophilus. You're bounded by your total number of sequences. If you ignore that, you get spurious correlations and your components appear dependent, even though they might be independent. This is a really good graph of that. It goes up, also has to come down. So you get these structural arcs here because of the compositionality in your data. And so it's just something we need to be aware of. There are methods to get around this and to work with it, but you can't just go use a PCA. So this brings us to our ordination side. PCA is the most common method for ordination in classical statistics. It's not really appropriate for microbiome data. You can use it if you do things like central log ratio transform, which attempt to normalize the data, but it's still not amazing. So we prefer the PCOA, which allows for different ecological distance measures. Something to note is that a PCOA and a PCA are the same if you're using a Euclidean distance. The difference is that a PCOA doesn't. It uses different metrics. And so things we think about that are the Jacquard, Bray-Curtis, Mauricio Horn, which allow for a little bit more flexibility when it comes to the compositional nature. And then I've also listed the NMDS. This is what you would get from a proximity matrix from something like a random forest. So that's another ordination technique that you can use. So again, considerations, we're trying to talk about the most variants explained in our data using all of our taxa instead of having to individually split them up and talk about them one at a time. Because again, we can't put them all in the same model. They're all correlated and you can't put 1,000 things in one model. Not good. But you need to be careful of the structure arcs. And you also need to be aware if you have repeated measures. Something that people have done with ordination methods is to make long data and then include all of that into the PCA. So long data would be, you have one subject and then sample 1, sample 2, sample 3, all from the same subject. But what that does is it, one, violates the assumption of PCOA, which says that your observations are independent. And two, it hides any real group differences that you might have, because in your microbiomes especially, you will look most like yourself throughout the progression of time. And so when you do that, you're missing real effects because you're just going to get clusters of individual subjects. So things to keep in mind. So then the next part of this are the individual taxa comparisons. So for this, the first thing we want to do is normalize, because like I said, each person has a different amount of sequence counts. So Bobby might have 30,000 sequence counts and Susie might have 1,000,000 sequence counts. And if Bobby has 15,000 counts of Pseudomonas and Susie has 15,000 counts of Pseudomonas, do they both really have the same amount of Pseudomonas? No, Bobby has a lot more Pseudomonas relative to himself than Susie. So we want to account for that. And there are 3 ways to do that. One is just a simple calculation of relative abundance, which is your count divided by your total sequences. So you say 15,000 divided by 30, 50%. Another way to do it is through rarefaction. This is what's being done in QIIME. And basically what it does is it randomly selects the smallest number of sequences and then it uses a simple calculation to pull out the ones that are the least abundant and kind of remove them for the variation. And then another way to do it is through a model-based approach. This is what's being done in things like EDGAR and DESeq, if you've used this. So it uses a distribution and it says how different from theta are we, and if we're too far, then we remove them. So those are things to keep in mind. I like RA, it's simple. It doesn't account for the heteroscedasticity, but I mean, there's pros and cons in both, and it's the simplest to explain. The next thing we want to do is filter again. This time we are not filtering for quality. We are filtering to determine which taxa we should actually be testing. And the 2 things we're looking at here are prevalence and then that relative abundance that we just talked about. So for prevalence, if you only have one person who has that taxa, you can't compute a variance or an estimate for the total relative abundance and then talk about it because only one person has it. So we want to remove those taxa. The other thing we don't want to do is we don't want to test taxa where the median for these is 0.000000001. Because what is the clinical difference between 0.0000001 and 0.0000015? I don't know. it's not much. And so we want to take those taxa out, and it helps with keeping down the total number of tests that we're going to do. Importantly, we are filtering at the taxa level, so we are not filtering individual subjects. If Susie has a low relative abundance, we're not just removing her. It's a whole taxa that's being removed. The other thing is we want to leave those taxa in for the overall community compositions that we talked about earlier. So we're not filtering until now because we want to know about their richness and their evenness and those diversity metrics together. And we can't know that if we've removed them. Everyone is frozen, and I'm going to keep going. So the first way to kind of think about this is just a simplistic approach. Again, said the microbiome data is that we have small sample sizes. So we don't want to go throwing the craziest models at stuff that only has 25 samples. So we want to think about things like visualization and descriptives. Great visualizations are things like this bar chart here, where we're saying, what are the most abundant taxa and are they different between our groups? The other thing is we can do what is similar to a t-test, but for non-parametric data. So we think about things like Wilcoxon rank sum and Wilcoxon sign ranks for paired data. And we can kind of assess the differences that way. So we could say, here's my stacked bar chart, and do I look different, yes or no? And that's kind of the extent we can draw there, but it's great when we have small sample sizes and when we're just trying to determine differences in community composition. So let's say we have a large sample size and we want to go a little further. We want an estimate around those groups. Things to remember. Taxa are right skewed. We have a lot of zeros. It's zero inflated, it's right skewed. They often can also look like a U shape, so they'll be bimodal at the tails. And basically what happens here is we're saying if you have the taxa, you have a lot of it. And if you don't have the taxa, you don't have it. There's nowhere really kind of in between, so you get these U-shaped distributions. So the other part is that relative abundance, again, is bounded between 0 and 1 because it is a proportion of the total sequence counts. So things that we do for this are we often think about negative binomials and beta binomials. And then I kind of listed out what that notation looks like. But also, because we have all of these zero inflated data, we want to think about zero inflated regression. So zero inflated regression basically just models the zeros on their own and then adds whether or not you have it to your relative abundance. So you're saying it's a prevalence on one side, do you have it, yes, no, and if you do, by how much? So these are the mixture distributions that we get into here. So we have zero inflated negative binomials and zero inflated beta binomials. The nice thing about these is that you can also extend them to random effects. So when you get into your longitudinal designs, you can use them. So this is a great graphic for the general approach to modeling. So over here on the univariate side, you have the Poisson, the negative binomial, and the zero inflated negative binomial, again, counts Poisson. And then over here, you kind of have the beta binomials and the zero inflated beta binomials. And then I listed over here the multinomial approach. So we talked earlier about how you have ordination methods where you're trying to synthesize all 1,000 together. And then this is kind of, well, what do we want to synthesize groups of them, things we know we're interested in. You can use a multinomial Dirichlet, which we don't go into, but essentially it is an overall test that you can use. It's there. This is a great graph to kind of demonstrate how the beta binomial might look. So this is that U shape that we talked about, lots of zeros, but when people have it, they have a lot of it. And the beta binomial and the zero inflated beta binomial fit this fairly well. On this side, we have the negative binomial. So this is that right skew. What we see here is that the beta binomial and the zero inflated beta binomial kind of overestimate it, but the negative binomial does a pretty good job. So those are the kind of the modeling techniques there. So now we've done all of our modeling, and now what we have to think about is, well, how many tests did we actually do? Because if we have 1,000 taxa, and we're testing all of them, we can't just say, that's great, we're going to move on. Because we know that if we have an alpha 0.05, we have at least 5 false positives in a sample size of 100. That's kind of the whole game here. So we're going to adjust for this. I listed Bonferroni because it's kind of everyone's favorite and the one that they know. But for microbiome data, it's pretty conservative because it takes alpha and it divides it by the total number of tests done. So if you're doing 1,000 tests, it's going to be really hard to find anything. So instead, what we do is what's called the false discovery rate. It's often referred to just as an FDR, which ranks each of the p-values and then just keeps the most significant ones. So now that we've done all of our tests and we have adjusted for the fact that we did all of our tests, how do we look at it? This is a really great graph. I've said really great graph like 16 times, and I'm aware of that, sorry. But this is a good graph that's in Tidy Micro, which is a package I'll talk about later. And it shows each of the estimates for the different groups we were interested in by taxa. And so it lists it out. Here's the rate ratio. I believe this was a negative binomial. So it's the rate ratio for each taxa by group, and you can kind of see who is differing and whether or not it's significant. And then this is the Rocky Mountain plot, which was developed by Rachel Johnson, who works at CETA now, and I think is really great. This says, if I have a taxa that is significant, by how much? That's what these bars are. They're representing how significant. So the more significant you are, the further this bar goes up. And then if it's going up, it's positively correlated. And if it's going down, it's negatively correlated. And then you have that for each of the taxa. In this case, it's the phylum for the taxa. And then we have some other analytics of interest. If you're like, okay, I've done all of that, now I want to go further, you can think about things like machine learning, supervised classification and clustering, unsupervised hierarchical clustering. This is also your ordination methods are considered in this little block. And this here is a graph that if you've looked at the paper for the StatsFest, this is in there. This is a hierarchical clustering graph. So you have your heat map of your taxa here, and then these are your outcomes. And if they were associated, you'd see like big blocks of color. It's a good visualization to show taxa that may be correlated with your outcomes. You can also think about things like phylogenetic trees, network analyses, and again, that multinomial Dirichlet. Network analyses and phylogenetic trees are becoming increasingly popular in microbiome, specifically the network analyses. There's a lot going into that right now. So now some useful packages and pipelines. I talked about Tidy Micro. Tidy Micro was developed here at Anschutz. So if you're working with data that's coming from the microbiome labs here, either at Children's or with Dan Frank, so Dan Frank or Kirk Harris, they both put input into this package, and it was created by Charlie, and I think he's still here, I'm not sure, but it is meant to work with the data that comes out of here. The other one is Workflow for Microbiome Data Analysis. This is made from Susan Holmes's lab, who's kind of the big microbiome guru. She's one out there, like Rob Knight is the other one. And this is really great. This has a lot in network analyses and phylogenetic trees. Both of these packages will walk you through from like the start of an OTU all the way down through the total analysis into beta binomials, negative binomials, and the Holmes package will go a little further into the network analyses. It also goes a little higher up at the beginning of the pipeline because it will actually allow you to align your sequences if you have raw sequence counts that aren't already put together in a table. And then the last one is explicit. This is great for people who don't want to program, have no interest in it, don't care, they just want to look at their data. And so you can put a text file or a CSV into this software, and then you can look at the stacked bar charts. You can look at the Wilcoxon-Stein ranks. It'll make stacked bar charts. I think I already said that. The alpha diversity, and then also some heat maps are in there for you to look at. So that's a great project. All of these are free to download and use. These two are in R. This is a standalone. You don't have to download anything but this. And then there's many others. So I talked really fast, but I left a lot of time for questions. So a big thanks to the VAP Consortium who donated their data to StatsFest and have mentored me throughout the years, and also to CU Data Week. Lala and Jonathan put a ton of work into this, and we're very grateful for them. And if you want to stay connected with CETA, here's our Twitter and the LinkedIn. Yeah, so if there are any questions, I'm happy to answer them.
Thank you for doing this talk, Kayla. It was awesome. Yes. Okay, so I noticed there's a couple of our StatsFest attendees on the chat, and some of them were my students from the data management class. And so I was hoping maybe you could talk about kind of, for you, since you've managed this data and you know which data sets they're receiving, if you have any like top 2 to 3 tips if they were approaching some of our data management research questions, what advice would you give them?
For like the data management research questions as
For
far as
StatsFest.
for StatsFest. Okay, yeah. I think like for handling microbiome specifically, or just for in general?
Let's make it specifically to the data that we're giving them for StatsFest.
Okay.
Yeah.
Yeah, no, so I was thinking about the clinical data versus the microbiome data, but I think the big thing for like data management of microbiome data is that it is very large. So if you can run a script on a small number, or like pick your favorite taxa and run your script on that and make sure that it works and that everything joins properly before you try to join all of it together, you're going to have a much better time than if you're trying to just put everything together all at once. Also, taxa names are weird and they can get kind of, they're also very long. So it might be good to, if you can, split them up. So, and what I mean by that is like take the last part of the phylogenetic tree and say, okay, this is my genus, and I'm just going to work with the genus names. That's often easier than trying to work with the whole phylogenetic tree as a variable name. Yeah.
Does anybody else have any questions? For those of you who are attending StatsFest, you're welcome to ask questions now, or Kayla will also be there during kickoff once you've had a chance to actually touch the data and read it into whatever software you're going to be using. You're also welcome to ask her questions tomorrow.
I have a question.
Yeah.
So since it's really like, I'm guessing that the goal is going to be discovery, right? So I'm wondering about the study design. Is it going to be like a case control design, or is it going to be a cohort where we're going to have all of these taxa that we're going to look at in a disease that has a small prevalence?
You can decide. Because the way that we have it is there are many different outcomes. We were hoping that people would explore different outcomes that we didn't already. So the main outcome that we looked at was VAP. There are lots of things in there like ICU admission category and admit, primary diagnosis, all things we didn't really look at. And the other part of that is that for the discovery piece, we didn't look at the microbiome just by itself. Like, are there groups in the microbiome that we should be thinking about? Are there phenotypes that we didn't find or look for, really, because we were focused on a very specific question? So it's a little bit of both.
Thanks for the non-answer.
I was just wondering, did that answer your question?
Yeah.
But it really is kind of what you make of it. I think things of specific interest would be like the clinical data for specifically the antibiotics. How are antibiotics related to microbiome? That's a big open question right now in the field. Mm-hmm. So if you're on certain antibiotics, are you seeing other microbiota that aren't pathogenic or pathogenic?
So are we going to have to pick a research question ourselves?
No, I listed some.
Yeah, we have a list of research questions that are a little bit broad. And then the mentors are going to be advising whoever the participants are but the participants can decide to take it as far as they want to go. So we have research questions that are like, okay, maybe you're a beginner programmer and you just want to get your hands on actual real data. You can do some of these like PLOT score calculations, or it could be like, you want to do a crazy analysis, let's do it. So it's designed to cater to various levels of experience, so everybody can kind of pick their own adventure, as I like to say.
I know that one of the like I would call it intermediate tasks that the investigators were really interested in is just looking at visualizations of who was on antibiotics. So just, can we make a graph of who was on different antibiotics and at what times? Because we don't really know. We haven't touched it really. So that kind of thing itself would be really interesting, which is all clinical. It's very interesting.
Anybody else have questions for Kayla? Okay, well, Kayla, thank you so much for doing this talk. I really appreciate you taking the time and for preparing our data for tomorrow. But yeah, I'll see you tomorrow. And actually, we just got a question in the chat. Have you used Microbiome Analyst?
No, I've never heard of it, actually. I've heard of Metaboanalyst, but I've not heard of Microbiome Analyst. Is it in our package or a standalone software?
Yeah, so I think that it's the same developer for the Metaboanalyst.
Oh, cool.
Yeah, so I just wondering how, I use the Metaboanalyst a lot, but I just don't know how you think about this. But
yeah, I can't speak to that. I've never looked at it, but it's cool to know that